This is an image of human skin, and these mysterious structures are known as Rete ridges.
So one of the questions has been, what is it? Is it a vestigial structure? Do we even need it?
Rete ridges could hold vital clues to regenerating skin, and maybe help explain why it is that humans don't have fur.
When the process of publishing is, it made us rethink everything, and that made us feel that this was a very important study to talk about.
Rete ridges are thought to harbour the stem cells our skin needs in order to heal, while also helping structurally to hold our skin together.
It acts like Velcro to keep your top of the layer of your skin attached firmly to the bottom layer of your skin.
But despite these important roles, there's little known about them as they're hard to study.
And part of the reason that we didn't know a lot about them is because there's so few models that are easily accessible and ways to actually study it. Most of the understanding about how it works inside of us as humans is just unknown.
So Ryan and the team set out to find an animal with skin that most resembled humans to study the Rete ridges. Luckily, they did know a good place to start.
So I don't think it's a secret, at least among skin biologists, that pig skin is more similar to human skin. The biggest problem is accessibility to the pigs and affordability to work with the pigs, and then kind of also doing a little bit of the smelly work as well. I didn't really want to work with pigs either, even though I knew that, as well as the rest of the field knows, that pigs are the better model, because it's really tough to work with them. So I searched for everything else other than pig.
So they looked at every alternative: dolphins, monkeys, mice, naked mole rats, even grizzly bears. And the team assessed them to identify the one that was most similar to human skin and found it was
pigs. Which is not a surprise.
Not that there weren't some surprises. It turned out that grizzly bear skin was particularly similar to humans.
Grizzly bears have Rete ridges. I could not believe that at all.
Only a few animals were known to have Rete ridges, and grizzly bears weren't one of them. The team also showed that the Rete ridges were associated with thicker skin and less hair. Animals like dolphins had more and larger ridges than furrier creatures.
The lower hair density that you have, it seems like their higher propensity and the ability to actually create these stem cell niches.
But to model these human stem cell niches, the Rete ridges, the closest match was indeed pigs. In the pigs, the team found that Rete ridges form after birth, something which had never been known. They even found the molecular mechanisms of how they form.
This is a really important thing to know because it's like laying the blueprint, right? If you don't have the blueprint at a molecular and cellular level, then it's like chance trying to get something to regenerate.
Unlike sweat glands or hair follicles, Rete ridges form via their own mechanism, developing where sweat glands or hair follicles don't. This new understanding could help researchers figure out how to promote regeneration in the skin.
It means that a lot of our approaches for regeneration need to focus on the mechanisms for Rete ridge formation. Young skin, when we're really young, heals much quicker, and also it regenerates their Rete ridges.
By restoring Rete ridge regeneration, Ryan hopes that we could unlock ways to keep the skin young or even reverse the skin's aging process.
The biggest unanswered questions is, if you want to get human skin to regenerate, how do you get Rete ridges to regenerate? And so now we have that blueprint.
And when it comes to why humans aren't furry, while Ryan and the team did show there's an association between hairlessness and Rete ridges, it's not always the case. And it's hard to know if the ridges arose because of this or caused it.
I would say it's too soon to tell because we can't go. It'd be wonderful to take a time machine and be able to get a biological sample from a long time ago to see what that was actually saying. I think it remains unanswered, and hopefully a really interesting question that other scientists can then utilize this understanding to actually go back and really think about domestication, evolution, and the acquisition of unique appendages.